One second. Cool. All right. So hey guys, like Mike said, my name is Dalton. So I started the MD PhD program in 2015 at Northwestern. So that's the same time that Mike and Celeste both started. So I'm sure you guys get a lot of great talks about biology and how it relates to medicine, but I doubt you guys talk very much about the role of chemistry in medicine. So at the end of the day, the vast majority of doctors, they either perform procedures or they prescribe drugs to fix a given ailment. So knowing how drugs work and how to develop one is a really powerful tool if you're interested in medicine more generally. So the chemical industry is unique in that there are many, many different large pharmaceutical companies that need to hire new chemists every single year that either design new drugs or oversee the production of existing drugs. And then on top of that, all those medical prosthetics that are used for surgery, and pretty much every single piece of plastic that you've encountered throughout your life has been formulated at least some degree by an organic chemist. So if you're interested in chemistry, there are many ways to improve people's lives and make a great salary doing it. So today, I'm mostly going to be talking about the medical aspects of chemistry, just because that's what my research is in and that's what I'm most interested in. But my research is currently split between two projects, but really at the core, they're fundamentally drug discovery projects. Let's see if I can. Okay, cool. So I'm not sure where all of you are in your studies. So we're going to start really simple and then ramp up. And then it's Mike's fault that I'm asking a couple questions throughout the talk because he wanted me to ask questions. I'm going to start off with this. So generally, there are two different types of chemical bonds. So there are ionic bonds, and then there's also covalent bonds. So does anyone know the difference between the two?
You may get something in the chat after a couple seconds, Dalton.
Let me pull up the chat.
Yeah, and we can interrupt you if people add. If you guys have a question for Dalton or an answer for Dalton at any point in his presentation, feel free to interrupt him.
Interrupt me. Ionic have opposite charges and covalent same. So yeah, that's kind of the point. Yeah, so like ionic bonds, you're gonna have an exchange of electrons. So if you think of sodium chloride, the sodium electron is going to go to the chloride electron to make salt, table salt. But then if you think about like chlorine gas or fluorine gas, and they have the same electronegativity, they're going to be shared equally, and it's going to be a completely covalent bond. So methane is the simplest carbon molecule, and it consists of carbon and then 4 hydrogen atoms. So does anyone in the chat think this is going to be ionic or a covalent compound? Bueller?
You got a 50-50.
Yeah, that's right, Ryan. It is. Those are all covalent bonds. So if you were to get technical about it, you could look at the Pauling electronegativity of each of these atoms, and you'll see that carbon's got 2.4, and then hydrogen's got 2.1. And since they're really, really close, they're going to be a covalent bond. But that's besides the point. So actually, most carbon-based compounds exist entirely of covalent bonds. And then these examples show that carbon can make a single, a double, or a triple bond. And if you really had to guess it, like on a test or something, ionic compounds are things like salt. So they're going to have a positive part and a negative part. And organic compounds are all going to be completely covalent. So Linus Pauling was the guy who, in the early to mid 1900s, worked on the quantum mechanical description of a covalent bond, and that got him his first Nobel Prize. And then 1954, and then later on in 1962, he actually got his second Nobel Prize. It was the Nobel Peace Prize for his work towards the opposition of weapons of mass destruction. So he's the only person to have ever lived to win 2 unshared Nobel Prizes, and he was a chemist, and that's probably not a coincidence. Talking about what organic chemistry actually is. So if chemistry is the study of matter, organic chemistry is just the chemistry of carbon. And so organic doesn't mean that the chemistry came from Whole Foods. It means that it's based on carbon-based matter. And in fact, most organic compounds are made entirely of 4 different atoms. So carbon, hydrogen, oxygen, and nitrogen. And it's kind of interesting that all of these very unique architectures can be made from just these 4 simple building blocks. So then organic synthesis is the art of converting simple and cheap organic compounds into complex compounds that have medicinal or commercial value. So if the valuable compounds are things like drugs, does anyone have any idea where the cheap starting materials come from? Well, it's kind of a trick question, so I'm going to go ahead and say it. So organic synthesis relies on petroleum feedstock. So you guys probably living in Chicago haven't seen an oil pump jack, but they're a pretty common sight in Texas, and that's where I went to college. So crude oil is siphoned off the ground and processed into gasoline. And then the byproducts are purchased by a number of chemical industries that convert that raw feedstock into simple building blocks that chemists can use. So then a scientist like Celeste will perform chemical reactions on that material to convert it into something biologically active that can treat a disease, and thereby creating a drug. So most people don't realize that the matter that's in the pills that you take when you're sick is actually highly enriched in the carbon atoms that were the ones in dinosaurs. So that's kind of an interesting tidbit. So I want to talk a little bit about the sources of different types of drugs. So here's a pie chart showing all of the FDA-approved drugs from 1980 to 2010. So if we look at the bottom here, we have vaccines and we have biologicals, and together they comprise like 20%. So that means 80% of all the drugs that are prescribed in all of the world are actually small organic molecules made by an organic chemist. So I also wanted to point out the fact that most of these are natural product derived. So synthetic drugs, so not related to something that's natural from the earth, actually only comprise 30%. So the largest share of this pie chart is actually organic molecules that are directly derived from nature. And that's really what my research is based on. And there's actually probably a lot of compounds that are natural products, and then others that you would be pretty surprised as a natural product. So take aspirin. This is probably found in every single medicine cabinet in all of your homes. Very common drug, over the counter, is actually from the bark of a willow tree. It's a commonly used analgesic. And then we have something called Taxol. So Taxol is very commonly used as a chemotherapy drug. Is completely derived from nature, and it's from the Pacific yew. So actually there was a huge race to synthesize Taxol for the very first time because the Pacific yew was endangered. So it was a tree that grows in California. So there's a problem where people were cutting down the Pacific yew to get Taxol out of it to treat people with cancer. But we were really scared that we were going to run out because we were endangered, and what were we going to do once we run out? So luckily, a couple of organic chemists in the 1980s were able to synthesize it for the first time. So now we have an unlimited supply of Taxol. And then this one you probably heard of, tetrahydrocannabinol, which is from the cannabis plant, is often used in combination with Taxol for chemotherapy nausea. So how do drugs actually work though? So most drugs work by targeting an enzyme that's working too hard. So generally an enzyme will have different substrates. And what enzymes generally do is they catalyze a reaction to occur. So we have a red and a yellow substrate, and they're going to bind to the enzyme. And that enzyme is going to help a reaction occur to make this orange product. And then at the end of it, we get out our product and we get the enzyme back, and it works in the catalytic cycle. So the enzyme is able to turn over many, many, many different substrates. So in many cancers, there's a particular protein that's overactive. That's the root cause of an illness. So how could an organic molecule help to treat a disease? Does anyone have any idea? I got all day. No, I'm just kidding. There's two different ways. So one way you can think of is we have a organic molecule that looks a lot like the substrate that can bind into that substrate binding hole, and it can prevent the real substrate from getting in, and that's going to prevent that reaction from occurring. So if we do that, we lower the activity of that enzyme, and we can help to treat the illness if that's the root cause of the illness. So that's called a competitive inhibitor because it competitively attacks that substrate active site and pushes the substrate out of that active site. So another way would be a non-competitive inhibitor. So this kind of inhibitor would bind to the enzyme and then completely change the way that the active site fits in 3D space. So it changes the shape of the active site, and then the substrate can't fit anymore. So this is another way that to prevent an enzyme from doing its job. And if that enzyme is the root cause of an illness, again, this is a fantastic targeted treatment for an illness. So that's two general ways that an inhibitor can help with the disease that's driven by a certain enzymatic process. So is every single part of the inhibitor important for stopping the activity? Well, if you were to, I know these are just kind of like blobs that are drawn up here, but like if you were to look at it, then probably not. So there's a certain part of the inhibitor that actually binds to the enzyme. And in chemistry, we call that a pharmacophore. So a pharmacophore is the part of a drug that actually does all the work of the drug. So there might be other parts of the drug that help it be soluble in water, or make it not taste bad, or help it get across the plasma membrane. But really, there's going to be a core couple atoms in the drug that actually interact with the enzyme and make those strong connections that are responsible for that drug's activity. So this is just an example of a pharmacophore. So this is a ball and stick model of a drug. And then this is the 3D representation of some protein, I don't even know. But we can see that highlighted in the colors here are some key interactions with that molecule and the protein or enzyme that really help this drug to actually work. And it's notable that the other components aren't really so important. So if we were to make modifications here, or maybe here, or here, where those circles aren't drawn, those are non-pharmacophore modifications, and that probably isn't going to affect the activity of the drug that much. But we can use that to tune different properties that we want in a drug. So we want it to have a certain molecular weight. We want it, we definitely don't want something that smells really bad or tastes really bad, so patients won't take it. There's all different factors that you can modify to get what's called drug-like properties. So the only reason I showed this slide is because it kind of helps you understand a little of my research I'm going to talk about. So like I said, enzymes, do chemical work, and then a lot of drugs stop them from doing that chemical work. What I'm going to be talking about today are neurotransmitter receptors. So it's the same principle, but it's not really a chemical reaction. So here, the enzyme is a receptor, and the chemical work that it actually does is it opens up a channel that lets ions in. So there'll be some ions on the outside of the cell, and there'll be less ions on the inside of the cell. So there's going to be a thermodynamic drive for those ions to rush in, just like if you were to, open or turn over a bucket of water, and the water is going to flow out. There's going to be a tendency for ions to rush through down their gradient. But these receptors don't really work, and these channels don't really work unless something binds to them to activate them. So this is what this figure really shows. So here's the serotonin receptor, here's the ions, and then what happens is the serotonin will bind to the serotonin receptor, and then those ions can rush through. So in some diseases, like the one I'm going to talk about, schizophrenia, that channel's open too much. So the way that we fix that is to make an inhibitor, once again, a competitive inhibitor that can bind to this pocket and prevent serotonin from getting in there. And then that way, we can prevent this ion channel from opening, and we can maybe help fix some of the symptoms of schizophrenia.
Dalton, you have a question about what's 5-HT?
Oh, sorry, 5-HT is serotonin. I didn't say that. So serotonin is like the generic term for the molecule, but its structure is like 5-hydroxytryptamine. So a lot of scientists will write 5-HT instead of serotonin just for shorthand. It's just the serotonin. Any other questions?
Not right now. I think Sage may mention, I don't want to ruin her talk ahead of time, but I think she may mention serotonin later. We'll see.
Cool. I'm going to talk a little bit about it in like the
Yep.
next 2 slides. Okay, so since beginning my graduate work, like I said before, I kind of had 2 projects. So some of it's kind of like medchem, traditional drug discovery, and the other one is the synthesis of these complex molecules like I talked about earlier, kind of mimicking nature. So today, though, I'm going to talk about what's kind of been my side project, but ended up taking a while, so it might be a main project. It's the synthesis of this complex natural molecule called melphaladine A. And then, so this is actually it potently inhibits a particular serotonin receptor that actually might be important for schizophrenia. And I'll talk a little bit about why that's significant later. But first, I want to talk about a little bit more about serotonin receptors more broadly. So, the central nervous system is composed of the brain and the spinal cord, okay? So within the central nervous system, there are a lot of different neurotransmitters that are the tools to help neurons communicate with each other. So some of the neurotransmitters are like dopamine, adrenaline, glutamate, and then like serotonin. So serotonin is like the oldest one. It's the oldest and most widely conserved neurotransmitter in the natural world. So serotonin receptors are found throughout the central nervous system and actually throughout the peripheral nervous system, which goes throughout the rest of your body. So, and just to illustrate that how widely conserved they are, you can give an SSRI or like an antidepressant to a lobster and it'll actually change its behavior because lobsters have the very same serotonin receptors that humans do. So this might be a little beyond what you guys have learned in school yet, but serotonin receptors are what's called the G protein coupled receptors. So they have like all these transmembrane domains. They hang out in the plasma membrane and span it several times and kind of like make a wave. And that there's actually many, many different types of serotonin receptors. So as I've shown here, there's 7 different families from serotonin 1 to serotonin 7. And then among those, some of the families have multiple, members. So there's actually 14 different serotonin receptors. So while popular science often associates serotonin with like a general sense of well-being or like being happy, serotonin receptors have a wide range of functions in the central nervous system. And I'm just going to give you a couple examples to show you that. So there's this one drug called sumatriptan that acts as a serotonin inhibitor, and it actually is used as a treatment for migraines. So a lot of people don't realize that serotonin is one of the major components to causing migraines or treating them. So aripiprazole is marketed as Abilify, and then that's a serotonin antagonist that's used to treat schizophrenia and bipolar depression. So again, a totally different disease state. And then there's this compound called Zofran, which is a different serotonin antagonist, and that's used to treat certain types of nausea. So all of these very, very different things, from headaches to psychotics to nausea, these are all different. These are all regulated by serotonin. So, there's not really good drugs that target 5-HT5 or 5-HT7 down here at the bottom of the list, and that's really what my research is working on doing. So 5-HT5, like what is it good for? So it's expressed widely throughout the central nervous system, but actually we're not really entirely sure what 5-HT5 does in either disease or normal physiology. But even still, we have a couple of different clues that indicate that 5-HT5 is a really valuable drug target. So is it a particular part of the brain that plays a maintenance in circadian rhythm? So circadian rhythm is what dictates when you wake up or when you want to go to sleep. So it might actually be useful in treating sleeping disorders. And then if you were to inhibit that receptor, the serotonin 5 receptor, it helps to cure some of the problems that go along with schizophrenia in mouse models of the disease. And then they did a statistical analysis of clinical data in Japanese patients with schizophrenia, and they found that there's a mutation in most of those patients of their serotonin 5 receptor that correlated strongly with having a positive diagnosis of the disease. But as of yet, there's really no findings that follow up these clues, because we lack the chemical tools that are required to probe the function of this receptor. So that's where my research kind of comes in. So these are a series of natural products. Don't really worry about the structures. If I didn't have them up there, I wouldn't have anything to talk about. So they were isolated off the coast of Japan in 2016, and a scientist in Japan actually isolated them and determined their structures using a variety of techniques that we don't have to talk about later. Oh, hold on, I got a question from Ryan. Why is PS much bigger in schizophrenia cases? Yeah, so so this is a mutation, right? So this refers to a proline, proline, and this so that's a type of amino acid, and then S is serine, so that is another type of amino acid. So the idea is they found that this mutation, changing it from proline to proline cysteine or serine or serine makes for some reason makes the chance of having schizophrenia much higher, and it's thought that it's because changing it from proline to serine changes the conformation of the receptor that causes it to be active more of the time than not. So it's a more active receptor that can be treated by an inhibitor like this drug. Yeah, it's an increased frequency. So this is a bunch of different patients. Some of them have schizophrenia, some of them don't have schizophrenia. And then if you do a statistical measure of what that amino acid is at that particular site, it shows that schizophrenic patients are more likely to have serine, and the baseline patients more likely to have proline. Does that make sense? All right, cool, Ryan. All right. Yeah. So, anyways, so these compounds were isolated and they were sent off to the NIH to really test what they actually do. And melphalanine A, which is here with the blue, it was just super, super selective for hitting serotonin 5A, which is the one that we've been interested in. And it really doesn't hit anything else. And I just want to stress that this is super rare, that a small molecule can have really, really good selectivity for one receptor over all the other ones. Like the examples I gave above, I was giving a really simplistic examples. Like all three of those drugs, they hit a lot of stuff. I was just talking about the one they hit the most, but this one really only just hits one. So that makes it a very desirable target. So for that reason, we became interested in making this molecule so we can figure out the pharmacophore that's responsible for its action on serotonin 5A. And so like they discovered this off like some algae at the bottom of the ocean off of Japan, and they've never been able to find more of that particular strain of algae. So I have a couple milligrams of this in my freezer at work, but as of now, I have like the entire world supply. So luckily we figured out a way to make it so we can actually work on this as a drug target. So one of the ways that chemists use to synthesize a complex compound is to figure out how nature does it and then try to mimic nature as much as possible. So nature has had billions of years of a head start to figure out the best way to make a natural product. So mimicking how nature does it is usually a safe bet. So luckily, the biologists that discovered this compound gave us a couple of clues based off genetic data to kind of guess how nature did it. And then at the bottom here, I kind of show how I adapt nature's synthesis, which is kind of like an A plus B type thing, to do an A plus B type thing in it's a different but very similar way. And we don't really have to talk about the specifics of the synthesis, but basically I was able to make that A plus B product here. And then through a couple of steps, I was able to do the first complete total synthesis of melphalanine A, so we could now actually do some of that testing. So I only showed the last couple steps here, but I kind of want to stress that this took like 18 months of like 12 hours a day. And yeah, 16 steps. It was like a 1% yield overall. Total synthesis is a very, very challenging field when it comes to organic chemistry. And there's a lot of trial and error and a lot of making starting material But at the very end, once you get your natural product, it's a very rewarding place to work. So, and then here at the bottom, I just show, even when you do make a compound, you can always improve the synthesis. So I'm actually working on right now when I'm in lab, making the synthesis more efficient so we can make much more of meperidine A, so we can do a lot more biological studies to improve upon its efficacy for a schizophrenia drug. So really, the core of this project was 1, to make meperidine A, but then 2, to figure out what the pharmacophore was that was responsible for that really great selectivity we saw for serotonin 5 receptors. So I made a bunch of different analogs of meperidine. So some of it without this part, like this one. This one's missing this bottom left part, and then this one's missing that northwest fragment. And then a structurally related natural product called dopargamine. So this one is missing all of this, and this one has the 2 methyls. This one's missing everything but this. And then, this is a really truncated one. And then really, we can test all of these for activity. And, based off the simple lock and key mechanism, we should be able to figure out, based off the different activities of each compound, which of these fragments is most important for binding to the receptor. And then we can actually make it better through chemical optimization to make a longer lasting, more potent, and more stable drug. And that's really all I have to talk about. This is just a picture of me in my lab. They're all some of my best friends. Like I said, I work for Professor Carl Scheidt, actually up in Evanston most of the time. And I've taught a lot of really great undergrads. Oops, I did not mean to do that. A lot of great undergrads like Khalid Hemmings, who's an undergrad at Northwestern. And yeah, I'll take any questions you guys have. That's all I got for today.
Thanks, Dalton. Any guys have any questions for Dalton?
Thank you. Does anyone in the chat
have awesome? That's pretty good.
Thank you, Ryan.
My lectures usually don't get an awesome, so you're doing well. No. Is 1% high enough for you to actually use it as an overall yield?
Yes and no. Like most natural products are synthesized at like 0.01%. But the idea is that you would figure out what simple component is actually important for its activity, and then you do something in 5 steps and it'd be like 50-60% yield. So yes and no.
I have a question. Has anyone done like a 5T, like the HD5A knockout in mice and see what happens?
Yes. So 5HT5A knockouts in mice are kind of complicated because mice have 5HT5B, but humans don't. Because the human locus has a nonsense mutation. So we just don't have that receptor. So they have another serotonin 5 receptor to rely on, and we don't. So it's not really like an apples to apples comparison. It's much more apples to orange. But yeah, a lot of the work right now is just knockout experiments. But then again, you don't really know, is 5B, should you knock out 5A and 5B? Should you knock out just 5A? So it's much more straightforward to just pharmacologically knock it out in a different mammalian cell line.
Gotcha.
Anybody else? Sorry, I probably went on too long. You probably are all asleep.
Ryan's awake at least, so that's one person awake. How do you think this is going to tie into your future career, Dalton?
So that's a good question because I don't know what my future career is going to be.
I'm aware.
I am really interested in psychoactive drugs and natural products, but most of my thesis work is actually on like cancer research. But then again, I'm writing this proposal for myocardial infarction drugs, and I think I like that too. So I really have no idea what I really want to do. I think that's really one of the draws for doing a PhD in chemistry, is that you can use that same toolbox to do all kinds of different things, and you're only a couple months of reading away to actually start a project that could end up in maybe a year or two in a very useful chemical probe for the experts in that field. So there's all kinds of different opportunities for collaboration. And, you know, my boss has no formal biological training, but we have ongoing projects for brain tumors, myocardic heart attacks, 2 different types of cancer. And you don't really need to be an expert in the biology to make a meaningful contribution from the chemical side, which is why I don't know what I want to do.
I thought you were going to say you didn't know your biology, but
yeah, that's it.
And you were a chemistry undergraduate major, right?
Yeah, I ended up being one. So I went into school pre-med, and I was a biochemistry and psychology double major. And psychology did not last past like the first semester. So then I decided I could still pull the double major card by just doing chemistry and biology, which is basically what I was doing anyways. So I got a degree in chemistry and biology, but I took a lot more chemistry classes, and I worked in 2 different chemistry labs in college. And you know, that I think that my research experience at the very beginning of college kind of led me to what I'm doing right now, for better or for worse. So think hard about that first job in college, because it might influence you.
In my case, it scared me away from biochemistry. So
That's good.
I mean, I was gardening basically for my first job, doing research in corn. So
In corn?
yeah,
What made you leave corn?
it's a bit different. I think perhaps the pace.
But like genetics, or you doing like corn, like maize genetics and stuff, like
No, more like cell biology. So we were studying cell
What is that
division.
lady's name? McClintock or whatever?
Barbara McClintock.
Barbara McClintock.
Yeah, she got her own stamp.
I have her stamp.
She got her own stamp for working on corn. You should have stuck with it, Celeste. You would have had a stamp.
All right, any other questions for Dalton?
Yeah, I'm just being a distraction now. I'll see myself out.
All right, thanks, Dalton.
Thanks for your time.
All right, peace, guys. Study chemistry. Bye.
All right, is Sage here?
Yeah, hi. Sorry, I continue to not have my own computer, so we're working with it. So I am going to cover a little bit about anatomy because we had a specific request for anatomy. So let me share my screen. I just now added a couple slides on serotonin specifically, just for you. Okay, so if you guys have any questions, please let me know. Hold on. Why can I not see you?
I can interrupt you, Sage, if we get a chat question.
You can interrupt me, but I also, is it half screen for you guys too? Or is it presenting? Okay, is it presenting now?
Yeah, now it's presenting.
All right, cool. So we're going to do just a little bit of neuroanatomy, and I will try to move through this as quickly as possible. Just so everyone knows, some of these slides are going to contain pictures of actual dissected human brains from donors. So just be warned. Okay, so before we talk about anatomy, we have to talk about the planes of anatomy. Super fun stuff. It matters a lot in the brain though, because humans stand upright and basically all other animals don't. So the way that their spine connects to their skull is in a single line, whereas with people, there's a curve. So you'll see dorsal, which is like, think of like a dorsal fin for a dolphin, is this top part of your head, but it's also your back. And so that plane actually curves like this. And then, so that's superior or dorsal, and then inferior or ventral is like your chin side, but also your chest side. And then anterior and posterior. Posterior, I think, should be obvious. And then anterior is like the side of your nose. And then also your chest side. So that can be a little bit confusing. And then the side to side is medial is more towards the center and lateral is more towards the outside. So those are the terms I'm going to be using. Please stop me if I get confusing to people. And then this is just sort of, if you cut the brain in the middle like this, it looks like this, and that's called a mid-sagittal section. If you cut the brain coronally, so that's like, imagine if you were wearing a crown and you wanted to slice down across your face and cut off the front of the crown coronally, and then the horizontal sections are cut this way. And we're not going to look at a lot of those, but and then this is a spinal cord section. Okay, central nervous system. So Dalton talked a little bit about central nervous system, and I think it's important to know that you can't really talk about the central nervous system without talking about the peripheral nervous system and what the difference is between those. So here the CNS is in orange and the peripheral nervous system is in yellow. And how do you sort of define the difference between the central and the peripheral nervous system? So basic rule of thumb, central is brain and spinal cord. A second rule that people try to apply is whether or not it's protected by something called the blood-brain barrier, which just means that not everything that's in your blood can cross into your brain and spinal cord. There's actually a very sort of strict cellular barrier that prevents the crossing of a lot of molecules and poisons and immune cells and various other things. And so the peripheral nervous system is outside that protective barrier, and so they can be attacked. So that's sort of another way to look at the difference. Just as a fun fact for the medical students in med school, we're taught that cranial nerves are central because they derive directly from the brain. But if you use the blood-brain barrier rule, they are peripheral. Okay, here's the brain. This outside part is called a meninge, and it's kind of like a protective covering for the brain. And then these are blood vessels that sit in the sulci, which are those little rivets in the brain. And then, so yeah, so basically these are the two parts of the central nervous system, the brain and the spinal cord. This is the only time I'm going to talk about the spinal cord, but these are the parts that if you run your hand along your back, you can feel little poking part of your bones, and that's this part of the vertebrae. And so much deeper to that, you have the actual spinal cord, and the spinal cord itself is a bundle of nerves, a very, very well organized, complex bundle of nerves. And then there's the disc. So if you have ever known anyone who's thrown out a disc, this can sort of burst and impinge on the cord, and that's or these nerve roots, and that's how you get pain from that. But yeah, so the spinal cord is a very important part of the CNS that's often neglected, but I'm not going to talk about it for the rest of this presentation. So I'm going to continue neglecting it, just like everyone else.
I have a question about white versus gray matter. Are you talking about that later?
Yeah.
Okay.
I'm not, but I can talk about it here. Actually, the spinal cord is a great segment to talk about that. So the biggest difference between white matter and gray matter is you can kind of think of gray matter are the cell bodies of neurons typically, and white matter is the axons of neurons, which is sort of these long projections that carry the action potential, which is a form of electrical information across a great distance. And the reason the white matter is white is that the axons are wrapped in a sheath called myelin. And it's sort of like how if you were to run a wire for a long way, you would insulate it with something. The same concept, so it's like insulation. And so in the spinal cord, you can see there's this little butterfly shape, that's gray matter. And then all this lighter part is actually lighter if you really dissect it, and that's white matter. And so all these white matter tracks are actually information being carried from the body up to the brain, or from the brain back down to the body. And then the gray matter are sort of the cell bodies of what's called interneurons, which means they communicate between two other neurons. And they tend to have sort of short distance effects. And other neurons will have their cell bodies here, and then the information transmission will sort of happen in the white matter track. Okay. So some of this talk is going to focus on the suprachiasmatic nucleus. So the thing about neuroanatomy that can be really frustrating when you're learning it is that the anatomy was all named before any function was assigned to any part of the brain. So the anatomy is basically all telling you where exactly something is if you were to cut up a brain. So this is the human brain, and we're looking up at it. So like this would be the bottom of your face. And this is called the chiasmatic nucleus. Actually, I think I have that, yeah. And what this actually is the crossing of your two optic nerves. So if these weren't cut, they would actually extend out to your eye. And the retina of your eye is actually considered part of the brain as well. And so this is the chiasmatic nucleus. So when we talk, so the suprachiasmatic nucleus is just above, just means above the chiasm. And so this is the optic chiasm, and this is the suprachiasmatic nucleus. You'll just have to trust me. If you can't see it, these differences can be really hard to see. And then the triangle I've put here is the hypothalamus. And again, this is just named because this is the thalamus, and the hypothalamus is below that thalamus. And the suprachiasmatic nucleus is a small nucleus within the hypothalamus. Okay. So why is the SCN important? So the SCN, which is what I'm going to call it from now on, because it's easier to say, is mainly involved in the circadian rhythm. So it's a major relay station for the various components that give you a circadian rhythm. And a circadian rhythm, sort of colloquially, is used to mean the natural waking and sleep cycle of any given animal. So humans are diurnal, that means we're awake during the day and asleep, or awake during daylight and asleep during nighttime. And so the SCN receives information about photoreception. So it receives information about light, and it actually can receive information about light even in the vast majority of people who are born blind are still able to transmit enough light information to have circadian entrainment. So that means they don't have trouble waking up in the morning and going to bed at night, which you might expect. So there's like information about light, and then it's also involved in homeothermy, which means maintaining your body temperature. And in animals like lizards and snakes that don't maintain their own body temperature, it's involved in locomotion. So if they get cold, this area gets very inactive, and then they stop moving, and it's one of the ways they slow down their metabolism. And then, so another way that this brain region functions is to be involved in metabolism. So the hypothalamus, one of the major functions that it has in general is regulating hunger. So that's one thing, but it also has outputs through the brainstem into lower regions of the brainstem that can control heart rate and breathing rate. And so that can also control how sort of alert you are, in addition to controlling how much sort of, how much you're calorically burning off at any given moment, in addition to keeping you a certain temperature. And then the last thing it's going to be involved with is stress. This was originally determined by, you may know that if you don't sleep for a whole night, or if you sleep very little for several consecutive nights, you will become irritable and on edge. And when this was studied in animals, it was found that just altering the function of the SCN is enough to induce that sleep-deprived state. And so it's really that not ignoring your own circadian rhythm can cause a lot of problems. All right, so the thing about neuroanatomy is that I can point to this all day long and it's completely meaningless. And the whole point is to know all of its connections and what it's actually doing in the body. So I'm going to talk a little bit about postsynaptic regions. So that means this one region in the brain is going to project all over the brain. It's going to have neurons in it that send information from what it has gathered from the eyes and from other organs and from what it knows about its own body temperature and metabolism. And it's going to send that information all over the brain. And so postsynaptic means after the synapse. Hold on, I can have the chat open. Let's see.
You have a question about eyes seeing upside down. I think that's more of an optics.
I am not going to cover that today. I can cover that on a later day, or I actually have a classmate who studies vision. But yeah, that's it. So the reason I'm limiting my neuroanatomy coverage is that there's a lot going on in the brain. But yeah, if there's time at the end, I can absolutely try and address that. Okay, so postsynaptic just means after the synapse. So that means after the neurons that came from the suprachiasmatic nucleus, what comes after. So one major brain region that the SCN projects to is the hippocampus. Does anyone know what the hippocampus is famously involved in? Memory, yeah. So there is the most famous neurological patient of all time. Patient HM had bilateral hippocampal lobectomy or temporal lobectomy. And so the hippocampus means seahorse. So I guess the first person that dissected this thought that this looks like a seahorse. And it has this little cinnamon roll shape if you cut it coronally. This is an image. This is like from an fMRI image. And so the hippocampus is famously involved in memory. So I want you to think about, as I sort of go through what the SCN is projecting to, if it makes sense to you or not to find major projections from an area that's really important for your body to know if it's time to be awake or alert or asleep, to project very heavily to these various regions. So the hippocampus, like I said, like Jamila said, is famously involved in memory. It's also famously involved in spatial mapping and coordination, which has been studied very heavily in animals. Okay, the amygdala. So this is an fMRI, which is a functional MRI, and you can see it's not as crisp. And the amygdala is, has anyone ever heard of the amygdala and knows what it's famously involved with? No. Okay. So the amygdala most famously is involved with fear and fear response, but it's actually been linked to all kinds of emotional functions. And so back here, I don't know if anyone, you can see that there's also a lot of things happening back here. And this is from a study where people were looking at faces displaying neutral or negative emotions like anger or sadness. And so it activates the amygdala, but it's also activating this area back here, which is visual cortex. So this was an expected finding. When you ask people to look at images, you get visual cortex, and it's a good positive control that they're seeing what they expect to see. But yeah, so this is roughly where the amygdala is. It's a very small area, and so dissection images are not that helpful. So the amygdala is like major emotion and arousal circuitry, and then the cerebellum is also affected. So the cerebellum is this little part of the brain that kind of hangs off the bottom of the brain, and often when people draw the brain, they forget to include it. It's really important, and it's really pretty and very different looking than the rest of the brain, and it's considered to be part of your quote-unquote ancient brain. And the reason it's considered that is that other animals, like reptiles that are phylogenetically more ancient or conserved for a longer period of time, have cerebellums, or they have structures like this. And so traditionally the cerebellum is involved in motor coordination and motor tasks, and that includes language because of the motor component of having to move your tongue and mouth. And the cortex is the picture of me in college. The cortex is just all of the squiggly stuff on top, and specifically the prefrontal cortex, which is, yes, that's the real brain. I studied neuroscience in undergrad, and we had a collection of brains from donors for our program. So the prefrontal cortex is what's called is typically associated with what's called executive functioning, because in the very first famous case, which you will also hear 1,000 times, is Phineas Gage, who was a railroad worker who got a railroad spike right through his head, through his eye, and he lived, but it totally changed his personality. And so that was the first sort of indication that there was something in your head that was tied to not only sort of autonomic things like your heart rate and your breathing rate, but your personality. And so the prefrontal cortex, this is sort of considered your executive manager area. Okay, any questions about that? Do those areas sort of make sense for circadian function? All right, cool. Okay, so presynaptically, and this is a little bit more related to what Dalton was saying, is serotonin. So the major serotonin producer, if you were here for my talk, you saw a picture like this for dopamine. The major serotonin producer are the Raphe nuclei, and they're in the brainstem, and they project all over the brain. And one of the places they project very, very heavily is the suprachiasmatic nucleus. And it's actually been found, now that we can trace bundles of axons based on what they're carrying, so now we can look at, is this axon bundle carrying serotonin? We can see that there's major input in what's called a plexus, which is sort of like a thick net of serotonergic input right onto the suprachiasmatic nucleus here. And so this is just to show you on a real brain, the dorsal Raphe nucleus, which is the specific Raphe nucleus that projects there, is not a big thing. And it produces the vast majority of the serotonin that's found in the brain. So when you think about serotonergic drugs, you're really affecting the whole brain by affecting just a tiny little portion of it. So just because I studied dopamine, I figured I'd throw this in, which is this idea that serotonin and dopamine have both been popularized as the happy chemicals, and it's obviously a little bit more complex than that, and modulating both in tandem or opposingly will have very different effects. So that's just something to keep in mind. And then another thing that I think is important is there's actually a high proportion of serotonin and serotonergic receptors that are expressed in your GI tract and not in the brain, which means every time you take a serotonergic agonist or antagonist, you're actually affecting your GI first because you're probably taking it orally, and then it will go into your blood and cross the blood-brain barrier, which we talked about is a very strict barrier, and get into the brain, and then have its psychiatric effect. And so actually a lot of the off-target effects of serotonergic drugs have to do with the way that it affects your GI system, and this can be very uncomfortable for people. So, you know, we get excited about studying these very specific neurotransmitters and forget that there's nerves all over your body that also use these transmitters. And in this case, actually, it's even just other GI tract cells that use these transmitters. Okay, I think the next, yeah, all right, so if we want to, I had included melatonin because the SCN is related to circadian rhythm, but I can also end there, or if people have questions, or I can find you a picture of how the eye works and explain that. Thought?
Seems
How
like
does
it's your call.
Alzheimer's affect?
Yeah, it's 5 o'clock right now.
Okay. Yeah. Okay, how does Alzheimer's affect the brain? Okay. Alzheimer's, maybe it's easiest to explain this in terms of the findings of Alzheimer's. So, for those who don't know, Alzheimer's is a disease or disorder that when people are alive, what we know about the way it affects their lives is it causes disorientation, and it causes, actually, here we can go back to, let me share again. It causes disorientation, and it causes memory issues. And so early on, it affects the hippocampus. And so, like I said before, the hippocampus is famously involved in memory, but it's also famously involved in spatial mapping. And so that probably explains some of the disorientation. And so what happens is in an Alzheimer's patient is the first thing that anyone noticed was that people who died of complications of Alzheimer's or died with very advanced Alzheimer's, their brains were skinnier. And so what that means is all these places where the brain is like touching itself, and it's all fluffy and kind of mushed in together, there started to be huge gaps so that there would be more black all in here. And the ventricles, which is these little internal holes that normally house cerebrospinal fluid, they got really big. And so that was the first thing that they found. And then you may have heard of plaques. It's really hard to talk about Alzheimer's and not talk about plaques. So that's like the second big finding in people in brains that have had Alzheimer's, is in this area first, and then progressing actually throughout the entire brain. You get these plaques, and they're made from what we call A beta, and it's a misfolded protein. And because it's misfolded, it makes these conglomerate proteins. And so in a young brain, you will misfold certain proteins, and your cells will be able to clean them away. They'll be able to degrade them, or if they get expelled from the cell and into the interstitial fluid or the extracellular space, they will be cleared away by immune cells in the brain. Those are called microglia. But as people age, that system falls apart. And so that's one proposed mechanism for why these plaques form, is just that in general, it's known that as people age, these kinds of things are dealt with more poorly. The second issue with amyloid beta is that beta stands for beta sheets, and the beta sheets allow the protein to stack up on itself. And so it becomes, instead of just being a bunch of sort of what they call soluble protein, instead of being a bunch of little tungstate proteins that can be cleared away by microglia, it becomes these big sort of messy, gooey plaques that are really hard for the body to break down. And then the last thing is it seems to have some prion activity, which means that misfolded amyloid beta is in some circumstances able to go and misfold properly made amyloid beta. And so it progresses its own creation. So those are sort of the three things that help form the plaques. And these plaques are directly correlated with that level of brain degradation that we talked about earlier, where sort of the brain loses its mass and the neurons are dying, or these beautiful white fiber tracks are dying. But I think your question, if I can find it, was more about what causes Alzheimer's. And the truth is that no one really knows. And when I say no one really knows, I mean that when they've tried sort of stopping the production of amyloid beta, it doesn't necessarily prevent the generation of Alzheimer's symptoms. There's another protein called tau, which I'm not going to get into as much, that also has been implicated quite heavily in the formation of these plaques. And so there's a new idea that the plaques are more of a symptom that there's an underlying disease process and not actually causal to Alzheimer's. And tau is a really good candidate for the next sort of wave of trials. I guess sort of where the research stands now is that we know that the symptoms in Alzheimer's are caused by degradation to certain brain regions faster than other brain regions. So that's why the memory goes sort of first. But we don't understand everything, and neuroscience is actually still a very young field. So a lot of these questions are not really answered yet. But I guess the last thing with Alzheimer's is the newest data sort of shows that to really have a chance at treating it, we need to find a solution that would be more like a multivitamin that you take every day after the age of 50 that would prevent these changes from happening ever, instead of waiting for people to present with the symptoms. Because in general, with neurologic disease, by the time you present with symptoms, it's progressed pretty far into your brain. And so there's not a lot we can do. The end.
Do we have any more questions? Oh, here we go.
How does ALS impact? Okay. So ALS is a little bit different. ALS is more peripheral. Okay, so the peripheral nerves carry in them what are called sensory and motor nerves. So it's pretty obvious what that means. So that means that there's motor output, which means these nerves go to muscles and they tell the muscles what to do. And then there's sensory input to the brain, which means that these nerves also go like to the tips of your fingers. And if there's pressure or heat or something, that information gets brought back to the brain. And so these nerves are really long, and they're made by neurons that have a tiny little cell body here, and then a projection, a single axon that will run all the way down to the end of your leg or to the end of your arm or down into your heart or your lungs or wherever. And so but all the cell bodies are kind of in the dorsal or ganglion, which are part of which is here, this little root here. And so these long axons are susceptible to disease. And ALS is one of these types of diseases. And so what happens actually in ALS is that coating I was talking about before, the myelin sheath that makes things white matter. Let's see if I have okay. I don't know if you can see that this is the corpus callosum. It's a significantly lighter color than this. I don't know if that's visible to you guys, but that's because you're looking at these axons. They run sort of between the two hemispheres of the brain. So they run like as if they were coming out of the screen towards you. And so you're looking at the axons like that. And so that myelin sheath that I was talking about, the like fatty sheath that protects the axons, but also helps them carry electricity or electric information. It's an ion exchange, which I'm happy to go into at some other time. But and Dalton covered a little bit, but they help them propagate that electric information for very long distances. Classically, a meter long is the thing that you'll hear people quote. And so what happens in ALS is that myelin sheath, for various reasons, will start to degrade. And so it's no longer protecting those axons. And these axons are very, very skinny, and they don't have a lot of, like when you study a cell, it's crammed full of all kinds of organelles and everything. And axons don't really carry a lot of those organelles. They are not very sufficient on their own. They really need transport of basically everything from the soma. And so those axons start to degrade. And when those axons start to degrade, you end up with what you see in ALS, which is first motor impairment that affects the extremities. So people often, when they're first diagnosed, all that's happening is that they're having trouble writing or walking, dancing, running upstairs, things that involve sort of the very extremities. And then as the disease progresses, this will start to affect even the muscles that help you breathe, the muscles that help you eat, and the peristalsis, and swallow. And so the reason ALS is sort of really scary is this idea that your brain keeps working, so you're totally there, you're totally cogent. So unlike Alzheimer's, where Alzheimer's tends to be more painful for the loved ones of the person with it, because after a certain point, that person doesn't remember, and so can become sort of happy again. But with ALS, it's sort of the person who has it, you're totally awake, and then your body sort of starts to fail you over time. So that's why I don't know, you guys might be too young for the ice bucket challenge.
Oh, wow.
How old am I? So anyway, this was a challenge before the invention of TikTok that had celebrities dumping ice water on themselves to raise awareness and money for ALS. The most famous person that ever had ALS was Stephen Hawking. And if you've seen videos of him, you'll know that he had a lot of, so Stephen Hawking had a lot of
better stage. You're not that old.
I know, so sweet. Stephen Hawking had a lot of technology that helped him. So you may know that he wrote whole books by sort of being able to move one muscle in his cheek. But until his mid-20s, he was walking around fine. But I've gone sort of off topic. But yeah, so the way ALS impacts the nervous system is it causes myelin degradation, which results in motor neurons. and specifically in the motor neuron. So it doesn't seem to affect other neurons as much, which is part of why the brain stays intact.
Cool. So I think we should save eyes for a different time, maybe.
Yeah, that's fine. Yeah.
I think the reason
Vision is really its own thing.
vision
Yeah.
is complicated. I mean, the reason you see upside down is mostly an optics thing, but we can talk about that a different time. I think next time is Elizabeth talking. Have we arranged that or not yet?
She can't next time, so that's
Okay.
going to be
So maybe we can talk about
in a
eyes next
couple of
time
weeks.
or
Yeah,
something.
we can do eyes, yeah.
Do you guys have anything else that you want to hear about? I mean, we're happy to talk about eyes. I don't think any of us are going to become eye doctors, but possible. But we're happy to talk about eyes. Brain tumor, why brain tumors exist. I think we also, is Elizabeth going to give a kind of cancer overview, or would that help to give it before her lecture?
No, she's going to give an overview.
Okay.
I mean, it's sort of a different question. Cancer in general works by cells that multiply. Is the question that because neurons don't multiply?
Yeah. I think we can talk about cancers. I agree that could be a good question about why. I think a lot
How
of
brain tumors
cancers
work.
are not of the neurons, but
Yeah, they're not. Yeah.
so little things I remember from med school. Yeah, so I think we can talk more about cancers. We're going to have one of the other mentors who I think some of you have met, Elizabeth. If you actually came to Prism, you've met Elizabeth, and she'll talk about her research, which is more cancer based. And so we'll probably talk more about cancer, I guess, in 2 weeks ish, probably. So maybe we can talk a little bit about eyes next time. Is there anything else you guys want to hear about specifically? We already exhausted all of our more organic chemistry.
More organic, yeah. Oh man.
Neuroscience, okay. That'll make Sage happy.
There's too much neuroscience.
Meanwhile,
Steven's just bitter because he has to study neuro now.
That's true.
yeah, we can probably work in some more neuroscience. I mean, we could talk about cranial nerves and eyes or something like that.
I mean, we can talk about biochemistry, which is relatively related to organic chemistry. 'Cause in my talk, I
Maybe
did
more anatomy.
like some of the.
We could also do some more neuroanatomy if we wanted. Do you guys want to learn about neuroanatomy or just general?
Or do you want like regular? Yeah.
Like, do you want to learn about muscles and bones or organs or neuroanatomy? Like anything in particular?
The GI system.
And I think sometimes the anatomy lectures are a little bit hard to do. Like, we don't want to just like go through anatomy of the whole body because it's really, really boring. Just naming a lot of different parts on the picture. So I think at least we find it helpful to specific
conditions based on the neuroanatomy. All right.
Sounds like Sage is going to be talking more.
I can do like cranial nerves or something like that too.
Yay!
Yeah, so maybe we'll talk some
Actually,
more about
fun fact.
neuroanatomy. Yeah.
Fun fact that I learned when I was researching the SCN is that bacteria also photoentrain and they have circadian rhythm.
Yeah.
Yay!
I'm not
And also
interested.
memory based on blue light, but
We can also talk about.
What
like,
about the anatomy of gluten disorders? Okay.
Yeah, we can
Like
talk about
GI,
gluten
yeah.
Like
disorders.
celiac? We can cover celiac.
It'll be a sort of weird mashup if we do some neuroanatomy and gluten disorders next time. Okay.
We can
We
cut
could
Sam.
do-
It'll be the same thing.
Sam's back to clinic.
Oh yeah.
He'll
All
be
right.
defended.
I'm writing this down.
Yeah.
We can do like a section on genetic diseases affecting neuroanatomy and then tie in celiac with that. Celiac is genetic also.
Yeah, and there's a lot of other digestive diseases that we could also talk about. I mean, you could talk about sort of like lactose intolerance is kind of an interesting one because it can't be genetic fully because otherwise you wouldn't be able to drink mother's milk. But
It is genetic.
It could be late onset genetics.
it's late onset genetic, but it's not. Okay. I don't know anything about genetics. Can we talk about how height is determined? Oh God.
Yeah.
I think
Okay.
we
It
can
sounds like
talk
we need more
about
genetics and more
genetics.
neuroanatomy.
Yeah.
Can we talk
Because
about the
genetics-
heart, guys?
Genetics can be really interesting. Like some of those early
Genetics is-
experiments with flies are wild. So we can definitely talk about that.
Maybe I'll learn.
Yeah.
We can learn about Barbara McClintock too, because that was actually genetics research that she was doing.
That's true.
Side note.
I have a heart condition.
There you go.
There are
Yeah,
a lot
Heart
of
genetic
flatter.
conditions. There you
There are genetic
go.
heart
Congenital heart.
Yeah, maybe
We
we
could
can
do
talk
congenital
about a
heart
couple.
disease.
I mean, I don't think we're going to cover all genetic diseases in
No, there's
one
no
lecture, but maybe we can highlight some really interesting ones that have some kind of weird effects.
Yeah, so we can
Why
do
Why
McDonald's
McDonald's is
I think
bad.
this is a really important question. I mean, nutrition is certainly important. It's a little hard to lecture on because the experiments in nutrition are really hard to do. If I tell you you can only eat fish for the next year, that's a really hard experiment to have anyone actually agree to. So we do a lot of experiments in animals, but it's really hard to get human experiments that are well controlled. But yeah, I think we can talk about nutrition. We usually try to work it in because it's something that you guys can actually change if you want to.
there's some
And
interesting
that you can
work on
educate the people around you.
So we'll come up with something for next time. And then the time after that, we'll probably hear more about cancer. And then if you guys have other interests, again, reach out to Blanca or reach out to us and let us know. And we can try to tailor what we're talking about.